It's a great pleasure to be here today. I assume that the audience comes from various backgrounds and with quite variable knowledge of MEG, but I hope to bring something new to everybody in my lecture. So I will start by telling you about a little bit about the development of magnetoencephalography, MEG, into a cognitive and clinical tool. So a short glimpse to the history of MEG. Then I will continue to MEG's established role in neurological diagnostics in the current clinical practice. Then I will continue to some emerging clinical applications where I think MEG will get more space and room in the future, especially mild traumatic brain injury, with which we have been working ourselves here in Helsinki, and about memory disorders, which are, of course, a big emerging field in neuroscience in general. And then I will end with some future prospects in MEG research in general. So MEG provides an excellent window to a working human brain. As you know, neuronal functions are addressed at very different levels. One can focus on the molecules affecting neural signaling and on the connections between neuronal cells. Also, functions of single neurons can be studied with intracortical electrodes, but in humans, this can be accomplished only during brain surgery. When we want to study groups of neurons at the same time or their interactions, we need non-invasive neuroimaging. And MEG provides an excellent temporal and good spatial resolution for that. So history of MEG is still quite short. The first electrical brain responses of the occipital alpha rhythm were measured less than 100 years ago in 1929 in Germany with one electrode on the scalp. Well, quite soon after that, an English neurologist, E.G. Adrian, who was actually very fascinated by the possibilities of EEG, and also himself contributed quite a lot to the development of that methodology, wrote in his famous letter to Nature that EEG is apparently not enough. With the present methods, the skull and the scalp are too much in the way. We need some new physical method to read through them. And in these days, we may look with some confidence to the physicists for such an instrument, for it's just the sort of thing they can do. And indeed, the physicists did it, and first MEG measurements were conducted in 1968, also on the alpha rhythm of human brain. And only during the following decades, the different methodologies for structural and functional neuroimaging have really emerged. There are probably people in the audience who are not that familiar with the principle of MEG, so I will briefly go through them. So MEG measures the tiny magnetic fields produced by the electrical activity in the brain. For a strong enough signal that can be measured outside the brain, probably thousands or tens of thousands of parallelly oriented neurons need to be activated simultaneously in the cortex. MEG is very good in following brain activation, both in time and space. While EEG, electroencephalogram, also provides excellent time resolution, the spatial resolution of that method is badly hampered by the skull and scalp in between the sensors and the neurons. However, MEG measures the magnetic fields produced by the electrical activity depicted as the green lines in the mid-figure, and these magnetic signals are not disturbed by the skull and scalp, and thus also the spatial accuracy of MEG is very good. MEG signals are very, very small, so of the order of 10 to minus 15 Teslas. So in comparison, Earth's magnetic field is of the order of milliteslas. So the detection of these signals needs special superconducting squid sensors that are immersed in liquid helium at minus 269 degrees of Celsius. In addition to these special sensors, the measurements need to be conducted in magnetically shielded rooms. However, there is increasing research in developing sensors that could operate in room temperatures as well. and I will briefly go into those in the end of my talk. These sensors are likely to make MEG systems cheaper in the future and also more flexible without the need of expensive and complicated cryostatics. Finland has been a big player in the field of MEG for decades. MEG measurements started in the Helsinki University of Technology, now called Aalto University, in the beginning of 1980s, first with one-channel device. But soon a systematic development of multi-channel devices started, and in 1993, the world's first device with whole head coverage was introduced. As you can imagine, the first devices were not of much use for clinical diagnostics, as you needed to move the device around the head to cover any larger area of the brain. So the measurements were quite long and also very exhausting for the subjects. But the 122-channel device was the first device that made MEG also a clinical tool. And nowadays we have 306 channels in our use simultaneously. MEG offers multiple views to brain functions. First of all, we can look at local effects, as demonstrated here on the left side, for stimulus-locked auditory responses over the left temporal cortex. And we can follow the strength of the activation as a function of time. We can also study local changes in oscillatory activity related to a particular stimulus or a task. But very often we are interested in neural networks, the interactions of different brain areas, and we start to have more and more tools to measure such interactions reliably. For example, by studying the coherent activity between different brain areas, as is demonstrated on the rightmost figure. Very important part of today's MEG research actually relates to the development and utilization of different computational approaches for getting more out of the signals and of their interactions. Whatever imaging methods one is using, mapping of brain functions first in healthy subjects is naturally the basis for understanding the same functions in disease. MEG, for example, has been extensively and is still extensively used to map the basic sensory cortices. With MEG, we can follow both in time and space what happens in the brain when we are hearing sounds, or when we are looking at different visual stimuli, or, for example, when we are making motor movements, like on the right side figure. And this kind of information can then be later utilized also in studying the same functions in patients. Epilepsy patients form clearly the most important clinical patient group for MEG, similarly to EEG. So MEG's most established role in clinics is the pre-surgical evaluation of epilepsy candidates. We know that approximately 30% of epilepsy patients have seizures despite optimal anti-epileptic medication. It is currently well known that epilepsy surgery can be a very good treatment for reducing and even for abolishing the seizures. Still, epilepsy surgery continues to be underused worldwide, although recent studies suggest that even 74% of these patients get more than 50% seizure reduction after surgery. These patients are also often very young, and thus there's a lot to save as to the quality of life and to working years in these patients. So compared with EEG, the better spatial resolution of MEG can help in determining whether the patient has single or multiple epileptic foci, the precise location of these loci, and if there are many of them, also their temporal activation order. So these are all important things for the surgeon to know before the operation. Here, for example, is an example of a patient in whom MEG was used to map the epileptic spiking activity to the area marked with yellow in the rightmost picture. During the first seconds of the recording, you can see that there's a spiking taking place only in the right hemisphere, but then quite rapidly it spreads to the left hemisphere too. So in a few seconds, the seizure is already in very many places, and the surgeon needs to know where it started from. In addition to that, the surgeon needs to know how this area is located with respect to the important functional areas of this patient. So for example, here, additional functional mapping has been conducted for locating the somatosensory areas, CEFs, and auditory evoked areas, so that the surgeon can then try to not to touch those areas if possible during the surgery. For the best diagnostics, we use MEG and EEG together and very often combined with the patient's behavior. So for example, we can video record our measurements. So if the patient gets a seizure during the recording, we can combine the recordings with the actual physical seizure as seen here in this figure. So when there is changes in the curve, the patient's hand is moving as a sign of epileptic seizure. So these are our current practices. And we have a steady flow of these patients in our laboratory. So we have a very long clinical practice for using MEG in this country in epilepsy evaluation. So for us, it has become a routine for locating the epileptic seizure places in those places who do not have any clear structural lesion in their magnetic resonance imagings, but we also use it for diagnostics in patients who do not have clear findings in EEG. The numbers of these patients who have been investigated in our laboratory per year, they are not huge, but for example, they take a long time, so usually the whole day. So for example, our laboratory takes 1 to 2 patients per week all year round for these measurements. And for this, we serve the whole Finland and currently also Norway, because they do not have MEG services there. With respect to the size of the population of this country, we do 3 times the amount of pre-surgical epilepsy evaluation than the United States, and even we should do more of them. So this is clearly a field which is growing in the future as well. But epilepsy is not the only established clinical application of MEG. MEG has also been extensively used in preoperative functional localization for other causes, especially for brain tumors, for kind of building preoperative roadmaps for the surgeons. So for example, in this figure on the right, you can see a prominent tumor, and the colored dots point to important functional areas, here to auditory and somatomotor areas. So there's hand area, lip area, foot area. And these are areas which the surgeon wants to serve during the operation. And these maps can be combined with structural images shown here on the left, where you can even then find the corresponding vasculature to help the surgeon to navigate to the right place during the operation. Use of MEG for this purpose has somewhat been diminishing during the recent years, as transcranial magnetic stimulation has been partly replacing MEG here as a cheaper and easier to use method. But in the future, we are likely to go towards a situation where we more flexibly use several functional imaging methods side by side in these patients and try to benefit from the good sides of each of them. Like in this case, MEG as completely non-invasive and very comfortable method is going to keep its position in this field. In addition to epilepsy and functional localization, MEG is also increasingly used to follow development in children and also in its disturbances, for example, in preterm infants and in children with cerebral palsy, in order to learn about the related cortical changes and also the critical periods for optimizing the timing of rehabilitation, for example. And MEG as completely non-invasive and quiet methodology is quite easy to use even with children, as you can see from these pictures. I will now continue to the next part of my talk, from the established applications of MEG to their future use. So in all clinical practice, we are now constantly facing the questions of how to best utilize all the existing data. So in our field, we have the possibility to gain information both from our functional studies, but also from the brain structure and of the possible behavioral changes related to certain disease in the patients. We also have increasingly amounts of other types of clinical data in our use, for example, genetic data, and we should effectively utilize all these together. I claim that the clinical functional neuroimaging, especially MEG, is kind of at its crossroads. So for establishing new, highly meaningful clinical application for it, we need to informatively bring in all the possible data we have. So for that, we need advanced computerized approaches. We need increasing multidisciplinarity to understand all the aspects of these diseases and the signals and the data that we can bring together. And I think that we also need a lot of good quality imaging data banks or other ways to get reasonable amounts of these precious data to be used with different methodologies. And I will now go to a couple of examples where we are trying to use this kind of these ideas that I presented. And first of them is mild traumatic brain injury. So mild traumatic brain injury is a huge health challenge that affects yearly more than 5,000 people in Finland alone. These patients are often young, which makes the proper diagnostics and follow-up very crucial. On the right side figure, you can see what happens in 100 patients after traumatic brain injury. So there are the red and orange ones, which are bad ones. So those are easy to diagnose. But then there are the greenish and blue ones, who are the white ones. And they are the problematic ones. So most of them do recover really, really well. But up to 15 to 20% of these mild cases do suffer from long-lasting cognitive symptoms without any objective changes in their structural brain imaging conducted with CT or MRI. We have recently suggested that MEG immediately after the head trauma could provide increased sensitivity to the diagnostics of these patients. Here we have especially concentrated on detecting changes in the prominent cortical rhythms that give information on both healthy and diseased brains. So in healthy adults, alpha and beta rhythms are really prominent in the back of the head and at the somatomotor and motor cortices. But slow wave activity below 7 to 8 Hertz is only normal in newborns and it disappears in adolescence. So emergence of slow wave activity after puberty is usually considered pathological. So what we have done, we have run MEG very rapidly after the patients have hit their head. So we have been able to collect nowadays already more than 26 subjects. We start to have almost 100 subjects in our pool, but we have been collecting them quite rapidly after they have visited our emergency polyclinics. And what we have been able to show is that there is indeed pathological slow wave activity that can be detected in these patients when measured soon after the trauma. In our first study group, we had 26 patients, and out of those, we had 7 patients who showed clearly pathological activity, compared to only 2 in our 139 control subjects. And what was also good to notice was that this pathological activity mainly disappeared during the follow-up of 6 months, suggesting that these patients indeed showed acute neurophysiological alterations, but the prognosis was mostly very good. And we have been recently analyzing the exact same data with machine learning approaches in order to get more sensitivity to the analysis. And indeed, this seems to be the case. We found much more patients in our machine learning analysis from this subject pool, suggesting that this kind of methods could be used in refining our diagnostic approaches quite easily, and would help us in finding those who might be in increased risk of developing problems later on, and who might benefit from support and follow-up in the beginning of the period after the trauma. So we are collecting more patients, and we are doing actually at the moment also EEG measurements on these same patients, with the idea of trying to find whether we could actually use some kind of combination of MEG and EEG measures to really pinpoint the patients who need this kind of follow-up. Another emerging field for MEG is the memory disorders. So memory disorders are a huge burden to the society and to its individuals. And there's an increasing interest in using different neurophysiological approaches in predicting disease trajectories in different memory disorders. And actually, the first functional changes in Alzheimer's disease are known to be at the level of synaptic activity. So that is neurophysiological activity, which makes MEG and EEG kind of attractive methods for addressing those changes. And indeed, using MEG and structural MRI, our Spanish colleagues have recently shown very promising results. They calculated functional connectivity measures between different brain areas, and by utilizing machine learning, they were able to predict which ones of the patients who suffered from mild cognitive impairments, so not yet a memory disorder, but something already in that field. So they were able to predict those ones who continued to develop to actual clinical dementia and who stayed in a stable phase. And this is demonstrated on the right corner figure. So there's still clear overlap between the progressive and non-progressive patients, but I would say that these are highly promising results. And actually, we are now together with the Spanish group doing a bit similar kind of thing. So this is a large five-year European Union project funded by Horizon 2020 called AI Mind. This project has started this year, and with our Norwegian, Spanish, Italian, and Finnish colleagues, we have a very ambitious goal of utilizing neurophysiology, namely EEG and MEG, and artificial intelligence for developing tools for predicting the development of mild cognitive impairment into clinical dementia. In this project, we aim at measuring altogether 1,000 patients multiple times with EEG and MEG. And this is something that we could not do alone, but we need the other centers. And we will utilize artificial intelligence methods to find features of brain activity that would predict the disease trajectory And furthermore, the brain imaging measures will be combined with other clinical measures available from these patients. And these are measures that are already known to correlate with the disease, such as certain genetic makeup and cognitive functioning. So this is, I think, a very good example of a more high-risk, multi-center brain imaging study. But if it's successful, it will be of very high gain, taking the really increased burden of dementia in our society. A very great challenge in developing new clinical neuroimaging tools relates to the great variability we can observe in most of the cortical responses, even in healthy individuals. So this figure depicts auditory evoked responses in two completely healthy individuals. So perfectly normal hearing and other auditory functions. But still, their responses have very different strengths, very different shape, highlighting the difficulty in using these same measures in indexing any impairment or disease. So there's still a lot to learn about the brain responses we are measuring before we can really use them with the most efficient way. But we are trying to go there and dig into these problems. And I think that novel data science approaches can really help us here. And I will now show you some results from our recent study going into that direction. So we have recently studied using MEG data from more than 100 sibling pairs, whether we can find heritable features from the spatiospectral MEG brain response patterns. So we have MEG measurements from these more than 200 subjects, and we used machine learning for trying to find such brain features that would be highly similar between siblings, but would maximally differentiate the families from each other. So this figure is a kind of simple illustration of those findings, but it shows now two MEG spatiospectral components in which we could map siblings very close to each other. So apparently we were able to find features from the MEG data which were really similar in siblings with a similar kind of genetic makeup. And as a next step, we actually search for the genetic associations for these low-dimensional descriptions of brain activity. We had collected blood samples from all these subjects during the measurement, and we had sequenced the DNA. And with this kind of analysis, we were able to find links to genes with functions, for example, in brain development, suggesting that these kind of approaches may be informative even in rather small subject groups of a few hundred subjects in trying to figure out what are the underlying genetic factors in these measures that we are collecting from these subjects. We actually had a very interesting side finding in that study. When we looked at each individual separately, we noticed that the algorithm was very good in finding individuals themselves across different measurements and tasks. And out of 20 subject datasets, the algorithm found the individual's own data measured in another day or from a completely different task almost perfectly, even after 5 seconds of data. We thought that this was really, really interesting. So clearly, there is something really individual in our brain data that can be picked up with the right methods. And we have started to call this the cortical fingerprint. And we think that it may reflect the underlying neuronal network structure that appears very stable within healthy individuals. And it will be now very interesting to test this approach in different clinical populations in order to find out whether we can learn even from rather simple and short MEG measurements, some informative features that would, for example, differentiate between subgroups of patients with different kind of disease trajectories. And we are now testing this approach, for example, in our mild traumatic brain injury patients, as well as in Parkinson's disease, in patients with different stage of Parkinson's disease. And we will use this also in the AI Mind study that I mentioned earlier. I will finally give a brief introduction to completely, at the moment, non-clinical field of research, but as a good example of what we can now actually do with the present methods. With computational approaches, we can also go back to rather old research questions in our field to learn more. And one of such questions is whether speech is somehow special to our brains. So in our recent study, we approached these very, very old questions, whether speech sounds are truly special signals to our brains and they are somehow processed differentially from other natural sounds at the cortical level, or whether speech sounds are just one particular sound category among others, and that their processing just reflects the processing of particular acoustic parts within those sounds. To address this question, we used speech sounds originating from different kind of natural categories, like musical instruments, animal cries, tools, and so on. And we also used their corresponding environmental sounds. I mean, the actual sound of the musical instrument, the cry of the animal, or the sound of the tool as a stimulus. We first composed different kind of acoustic descriptions of these natural sounds. So we used the overall power distribution described by fast Fourier transform shown in the figure. And then we used spectrogram, which describes the power distribution of these sounds over different frequencies as a function of time. And last, we used the modulation power spectrum, here averaged over time, which describes the spectral and temporal modulations within the sounds, which are known to be important for the recognizability of natural sounds. We then aimed to learn the link between these auditory descriptions of the sounds and the cortical response to these sounds with machine learning. And we addressed the cortical responses either as a whole, that means averaged over the whole time course of the response, for those stimulus models that were not dependent on the time, that means the fast Fourier transfer model and the MPS model. Here we used non-time sensitive regression model to learn the connection between the sounds and the brain responses. For the time-dependent spectrogram sound model, we used a time-sensitive convolution model and assumed that the MEG responses followed with some delay the time points in the acoustic signals. And these models were then trained with part of the data and then tested on the rest. And here are the general results. So when we used those models that did not have any time information within them, then the spoken words and environmental sounds were predicted much better than by chance, but rather similarly compared to each other. But when we moved to the time-sensitive convolution model, where we had the time sensitivity taken into account, the predictive accuracy for spoken words increased radically, while no difference was observed for the other natural sounds. And we have interpreted this to suggest that speech indeed is specially processed at the cortical level, with kind of dynamic time locking to the sound which unfolds on time. And this is something that, of course, can be studied with only time-sensitive measures like MEG and EEG, but also using time-sensitive analysis methods in between. And actually, we are taking this approach also to the clinic in the sense that we are currently investigating whether children with language learning impairment actually would have a problem in these kind of processes at the cortical level. If you were Finnish, I would actually play you some sounds here, because we have also now gone backwards. So after we are able to find the connection between the sound that was heard and the MEG response, we can actually use the MEG response to build back the sound that was supposed to be there, evoking the response. And they are not perfect, but actually, if you understand Finnish, you can understand the word from there. But for you, it would be mainly nonsense. So I did not bring them here too. But basically, we can build these kind of connections already with our data. So in the end, I will still go back a little bit to the MEG technology. So neuroimaging in general has really revolutionized brain imaging. So it's less than 100 years ago when our knowledge of brain structure came from postmodern studies, and the functional imaging was based for a long time on these quite simple EEG measurements based on only few electrodes. Now, we can get a very precise picture of existing or emerging structural changes within tens of minutes after a patient has entered the emergency room. And similarly, we have several excellent functional imaging methodologies which can give very thorough picture of the brain functions. But the story has not ended yet. So the technical development continues also on the MEG field. And especially our university, Aalto University, is quite active in this field. And one of the emerging applications of which I think is very appealing is the hybrid MEG MRI device, which is currently developed in a European multi-center project headed by Aalto University here. So the possibility to have very high quality MEG sensors in the same device with low field MRI can potentially bring several clinical advances. For example, via better structural precision to the MEG measurements. But this is highly demanding. So as you have learned from my lecture, MEG is highly disturbed by any magnetic disturbances. So building this kind of device is not simple at all. But the progress has been very good, and the first measurements in healthy subjects have been conducted with this device. Another important line of development, which I already mentioned earlier, are the so-called next generation magnetometers, which would be able to operate in room temperatures. And there's a lot of development in this field as well, and the first prototypes are already in use. And these are expected to provide flexibility to the measurements. These sensors can be brought closer to the head, so that can give better signal to noise ratio compared to the current helmet-shaped machines. And these would be independent of the expensive and complicated cryogenic systems that we nowadays use in the universities and hospitals. So there's a lot to come. But this was the end of my talk today. So thank you for listening. And in the end, I also want to thank all my collaborators who have benefited for this studies that I have shown you today. And of course, my main funders. Thank you.